In this video, we're going to talk about the mechanisms of translation, and we're going to talk about the different type of mutations that exist and how they might affect the structure of a protein. So when looking at the steps of translation, we can break it down like any of these processes in molecular biology. We can break it down into three components: initiation of translation, so getting everything set up; elongation, so actually adding to the polymer; and then termination, ending translation. Let's take a look at initiation. In initiation, what we have is the small subunit. Remember, the ribosome exists separately. The two subunits exist separately in the cell, and they only come together when there's mRNA involved. So the way this starts is the small subunit binds the messenger RNA such that it scans starting from the 5' end until it reaches the first AUG in any of the three reading frames. At that point, that's where our initiator tRNA, right? It's carrying methionine. Therefore, we know that its anti-codon recognizes an AUG codon within the messenger RNA. At that point, the large subunit recognizes this structure and binds to it. So here we have the initiator tRNA straight into the P site, right? It bypasses the A site, straight into the P site. It's bound to that first AUG starting from the 5' end. Doesn't matter what reading frame that AUG is in. And now that is the end of initiation. And we now move on to elongation. Now, the first of the three steps of elongation is that the incoming tRNA recognizes it has an anticodon that is reverse complementary to the codon sitting in the A site. And it binds, and we now have it sitting there within the A site. And we have hydrogen bonds connecting the anticodon of the tRNA to the codon of the messenger RNA. And the incoming amino acid is right here, and here, being carried by the previous transfer RNA, is the growing polypeptide chain. So now we go on to the second part of elongation. This is called peptide bond formation, where we get our catalysis occurs. The formation of the peptide bond that connects this amino acid of the growing chain to the incoming amino acid carried by this tRNA here in the A site. Now, this is a biochemical reaction that requires an enzyme to be sped up. Without the enzyme, this reaction is infinitely slow. And the question is, well, what catalyzes this reaction? And the answer is, globally, the ribosome, but more specifically, one of the RNAs that make up that ribosome. Remember, ribosomes are ribonucleoproteins consisting of both proteins and RNA, and it's the RNA itself within that ribosome that catalyzes the formation of the peptide bond. So at the end of this, what we have is that growing polypeptide chain now is covalently attached to the tRNA, the incoming tRNA that's sitting in the A site. And the tRNA that used to hold on to that growing polypeptide is now is still there within the P site, and we're ready to move on to the next step of elongation called translocation. And in this step, okay, so here's where we are, right? This is the same as what we were looking at. Everything, the ribosome shifts to the right, exactly one codon. What that means is this tRNA, this empty tRNA, is going to end up in the E site. That's the exit site where it takes off. This tRNA that is carrying the growing polypeptide chain is going to be in the P site. There we go. And then the A site is now open. We've got that new codon, that new next codon that is sitting in the A site, and that is going to be then recognized by another incoming tRNA, and we start that process all over again. Now, again, elongation is going to occur until within the A site is a codon that is either UAG, UAA, or UGA. Those are stop codons. Those are not recognized by any anti-codon. They are not recognized, right? They do not encode an amino acid. Instead, they're recognized by a protein called a release factor, right? So what type of macromolecule is a release factor? It is a protein. It is not a transfer RNA. And that protein recognizes these stop codons when they are in the A site of the ribosome. And that catalyzes the ending of the translation. It catalyzes the hydrolysis of this protein from the tRNA, so they are separate from each other. And there's that free polypeptide, and everything breaks down, and now that messenger RNA can be used again by more ribosomes to make more proteins. So it's a single messenger RNA is a template for the production of lots and lots of protein molecules. Now, this isn't an energetically expensive process. You don't have to memorize how much GTP is involved in each one of these steps. But remember, GTP is equivalent to ATP. It takes a lot of energy for cells to make GTP, and so it's pretty costly to then, therefore, spend it. So here we can see how a single messenger RNA is the substrate of multiple ribosomes, making multiple copies of that protein. And it happens at the same time. So we call this a polysome, where we get multiple ribosomes binding that RNA at the same time. Now, they all have to bind here at the 5' end. They all have to scan for that first AUG. But once a ribosome begins to move and goes into the elongation phase, another ribosome comes in. And we can see that here on this particular image. Now, let's go back to our discussion of cell biology in Chapter 6. And remember, we talked about the difference between smooth endoplasmic reticulum and rough endoplasmic reticulum. Well, what was it that made rough endoplasmic reticulum rough? Well, it was the bound ribosomes. Okay, so all ribosomes start free. All ribosomes start floating around the cytoplasm. And it's there that they bind that messenger RNA. Now, each messenger RNA has the ability, so some, I should say, let me wind that back. Some messenger RNAs will have encoded in them these first few amino acids that are a signal peptide. They signal to the cell, hey, I am a protein that needs to be made into the rough ER. I am going to be secreted, or I am going to be another protein involved in the endomembrane system. Now, the signal peptide, when it gets made, so here we have, this is a polypeptide that's being made. When it gets made, it gets recognized by another protein called signal recognition particle, or SRP. That SRP binds, recognizes and binds that signal peptide, and stops translation. Brings the entire complex to a translocation complex on the membrane of the rough endoplasmic reticulum. When it binds, it allows translation to then resume. The protein now is made straight into the rough ER. The signal peptide is removed enzymatically, and the protein then is processed in the rough ER, and then will be packaged up in a transport vesicle, and so on and so forth. So it's these signal peptides that tell the cell, hey, hold up, stop translation, this thing needs to be made into the rough endoplasmic reticulum. Now, once a polypeptide is made, it's not necessarily the end of the story. Many proteins, to be functional, need, for example, covalent modifications. So the addition of phosphate groups to some of the R groups, the attachment of sugars, the attachment of lipids, the attachment of various cofactors like magnesium ions and heme groups. Some must be properly cleaved. For example, remember, every single protein that is made starts with a methionine, but that doesn't necessarily mean that every protein requires that methionine. So in some proteins, they're cleaved off. Some proteins like insulin have to be, the insulin protein itself must be cleaved in order to properly fold. So lots of post-translational modifications must occur to some proteins in order for them to properly fold and function. Let's take a look at bacteria. What we just got finished talking about was translation as it occurs in eukaryotes, cells that have a nucleus. Now, bacteria and archaea, so prokaryotes, do not have a nucleus, which means the place in which transcription occurs, so transcription is reading this blue DNA template, writing this red messenger RNA, happens in the same place as translation, which is the ribosome making purple protein while reading red messenger RNA. And notice, because these two processes occur in the same place, they occur at the same time. So as soon as enough messenger RNA emerges from the RNA polymerase as transcription is taking place, ribosomes will pop onto there, because remember, messenger RNA is always made from 5' to 3', and so it's that 5' end that first gets made. And that's the same end that these ribosomes need to bind to in order to start translation. They scan, starting at the 5' end, for that first AUG, and that's where they start producing protein. So it's pretty efficient, this coupled transcription and translation in bacteria, in archaea, so prokaryotes, makes the production of proteins very fast, very efficient. Okay. So let's come back now. And the big picture of why we are describing transcription and why we are describing translation is because that is the method cells use to make traits, to make structures, right? We take the information in our genotype and all of the DNA of our nucleus, and we convert that to our phenotype, all of our traits. Right? The blonde hair, your blue eyes, your brown skin, whatever traits that you have is dependent upon reading the information within a gene to make messenger RNA, reading that messenger RNA to make protein, that protein properly folding to have all of its proper functions, and then therefore giving rise to your phenotype. Well, it turns out that the sequence of DNA in all of your cells is not, quote, written in stone. It is mutable, right? And what that means is if it does change, so here we have that fast acetaldehyde dehydrogenase gene, and we have the slow acetaldehyde dehydrogenase gene. Now the allele means, we'll get to that word a little bit later in the course, but allele is the different versions of that gene. Here's the version that has a C right here, and here's the version that has the T there. So the allele with the C and the allele with the T. Now that one change in the sequence of the gene causes a single change in the amino acid. In the fast enzyme, we've got the W amino acid, and in this one here, we have the S amino acid. So I believe that's a tyrosine to a serine. And although that seems like a subtle change, it's enough to change the structure of that protein such that now it doesn't work very well, which gives rise to a completely new trait. You have here a sort of normal response to ethanol, and here, if you have the slow enzyme, you have that alcohol flush sort of trait. Right, where when you drink ethanol, get red, flushed faced, and so on. We have another example here, where within the gene of beta globin, so here we're looking at the beta globin gene in DNA, and this is the normal sequence here that gives rise, right, GAG in the gene strand gives rise to GAG in the messenger RNA, which encodes a glutamine, a glutamate. And that's the normal hemoglobin protein. But if a mutation occurs to convert that A to this T, meaning within the messenger RNA, now instead of a GAG, you have a GUG, that now encodes a valine, and that changes the structure of the hemoglobin protein in such a way where they now stick to one another, they form these fibrils, and that distorts the red blood cells, and that causes all sorts of problems. And that is collectively called sickle cell disease. So how do changes in DNA sequence occur? They are called mutations. And the first type of mutations are point mutations, in which we are simply substituting one base for another, one base pair for another. So we're not adding to nor subtracting from the total amount of DNA. So let's take a look of the consequences, the possible consequences of these single base pair substitutions. If we take a look at the wild type, here we have our template strand up here. This is then below is going to be the gene strand. Notice how the gene strand is the same as the messenger RNA GGC, GGC. And then here is the protein that is going to be made from that messenger RNA. So the first possible consequence that can occur with a single base pair substitution, again, where we're not adding, we're not subtracting, we're substituting one base pair for another, is a silent mutation. And let's take a look at that. Here we have our wild type, GGC. And here we have a mutation in which instead of GGC here, it's GGT, right? So there's a mutation in which instead of having a C, it's a T. Since this is the gene strand here, that's going to encode a messenger RNA that is GGU. Well, we go from a GGC, which encodes a glycine, to a GGU, which encodes a glycine. So there was no change in the structure of that protein. Our mutation took a codon that encoded glycine and turned it into another codon that encoded glycine. Why are silent mutations possible? How is this possible? Remember, redundancy. There are many amino acids that are encoded by more than one codon. Glycine is one of those amino acids. It is not only coded by GGC, but it's also encoded by GGU. So it's possible to have these base pair substitutions that don't affect the protein structure at all. Again, because of the redundancy of the genetic code. Now, the next consequence is something called a missense mutation. So let's take a look. So again, we have our wild type DNA. This is the template strand that gets read. This is the gene strand, the mRNA-like strand that's going to look like the mRNA. Here's our first AUG, and then we go from there, methionine, lysine, phenylalanine, and glycine. And now what happens if we mutate this G here to an A? So instead of having a GGC, we have an AGC. Well, if you take a look in the genetic code and you look to see, well, what does AGC encode? It's a serine. So now we've gone from a glycine to a serine. That is the definition of a missense mutation. We have changed one amino, via this mutation, we've changed one amino acid to the other, to another. Now, whether or not that really affects the structure of a protein, well, it's obviously going to affect the amino acid sequence of a protein, but it may or may not affect the structure and function of the protein. It depends upon how similar in structure are glycine and serine. Are they both negatively charged amino acids? Well, in that case, maybe there won't be a big change. Are they very different amino acids? Well, maybe then that'll cause a big change and lead to a new function or a reduced function or a gain of function of that protein. And finally, nonsense mutations, where here we have an AAG codon within the mRNA-like strand, which of course is going to lead to an AAG codon within the messenger RNA that encodes lysine. Now, if a mutation occurs such that instead of having an A here, it's a T, so now TAG, which then is going to give rise to a UAG within the messenger RNA, that is now a stop codon. So we used to have methionine, lysine, phenylalanine, and glycine, but now it starts here, methionine, and then bang, stop. The cell doesn't know that that's not a correct stop. The cell doesn't know that, hey, wait a minute, you're supposed to make more amino acids over here. When it hits that stop codon, it stops, it's done. We end up with what's called a truncated protein. That protein is smaller than it should be. All right, those were the results, the possible ramifications of single base pair substitutions. Now let's look at what happens when we insert bases or delete bases. So we actually are subtracting or adding bases. Well, that leads to the possibility of a frameshift mutation. So let's say here we have a AT base pair right here, but there's a deletion that removes it. We delete this AT base pair, which means within the messenger RNA, we're missing this U. So we go AUG, that's great, methionine, AAG, that's lysine, but then UU, now it should be a U, but that's missing, but the cell doesn't know that. The ribosome doesn't know that. It just goes UU, well, it goes right straight to the next base. UUG, well, that's not the same as a UUU, so that's a different amino acid. And then GCU, well, wait a minute, that's wrong too. It should be a GCC, but we're now in the wrong reading frame. That's alanine. And so after that deletion, every single amino acid is going to be different than what it should be because we've had a frame shift. Okay? That's a pretty drastic mutation. It's also possible that a deletion or an insertion can cause a nonsense mutation, can cause a stop codon to appear where it should not appear. So if we add an A here, so now we have an extra U here, we go AUG, well, again, the ribosome doesn't know that this U shouldn't be here, doesn't know that it should actually read just AAG, but instead it ends up reading a UAA, which is a stop codon. Okay? So these insertions and these deletions can add to frameshift mutations, giving rise to lots of missense, and then it can also give rise to a nonsense as well, premature stop codons. Here's a little trick though. As long as an insertion or a deletion is a multiple of 3, then you regain the reading frame. You get back into the proper reading frame. So for example, if we have a deletion that deletes these 3 nucleotides, AAG, so the TA, the TC, the CG, this entire chunk here, then what happens is, okay, the ribosome binds AUG methionine, great. It misses this AAG because it doesn't exist anymore, but it puts us right back into the proper reading frame, UUU, phenylalanine, GGC, glycine, and so on and so forth. So as long as those insertions and deletions are in multiples of 3, the reading frame is going to be maintained. Now, obviously we are missing an entire amino acid there, or if it's an insertion, we will gain amino acids. That may or may not have ramifications to the function and structure of that protein, but at least we're not hitting a frameshift mutation, which causes a mess up of all the amino acids after the mutation. What are some causes of mutations? Well, many of them are mistakes in DNA replication. Right? When a cell divides and that DNA polymerase is reading and writing and reading and writing, it might make mistakes. Okay? Those are spontaneous mutations. There are chemical mutagens, right? There's base analogs. These are chemicals that look like nucleotides. They can get inserted into DNA, and that can mess up DNA replication. There are physical mutagens, right? These are the ones, the X-rays, or ultraviolet light. So if you are out in the sun, that can cause damage to DNA and can lead to mutations. Now, the ultimate question here is, will my children inherit my new mutation? So you will gain mutations to all kinds of cells, various places in DNA throughout your lifetime. It's unavoidable. But will that mean you're going to be passing on those mutations to your children? Well, It depends. It depends on what cells have that mutated DNA. Because when your DNA mutates, it doesn't mutate in every single cell of your body, right? If you were in the sun on the beach without sunblock over and over and over again, it's going to mutate DNA within the cells of your skin, not the cells of your heart or the cells of your liver or whatever, right? It's not being exposed to the sun. Those are called somatic cells. So any cell that is not your sperm or your eggs, those are somatic cells. Those are not going to be passed on to your offspring. Now, it could have problems for you, right? For you, you may end up with cancer or something like that, but it's not going to be passed on to your children. Now, if those mutations occur within germline cells, within your sperm, within your eggs, okay, and there's about 32 new mutations per person per generation that will appear in sperm and eggs. Now that mutation is going to be passed on to the offspring. Now, if that mutation causes an effect on a protein or something that leads to a new trait that is selected for in the current environment, then that'll be passed on more to more and to more offspring. If, however, that mutation is negative within the current environment, it would be selected against. And whoever inherits that will be less likely to generate offspring. And finally, some mutations will be neutral, that it will not have any effect that is either selected for or selected against within the environment. Now, most mutations cause no problems. Only about 1.5% of our generation encodes protein, and so the chances that a mutation will hit a protein encoding region of the DNA is small. Last thing we need to cover. I know that through these couple lectures, it seems like every single gene within the genome encodes a messenger RNA, which then encodes protein, but that's simply not the case. In reality, most genes encode RNA that is not translated into protein. Most of the RNA that's made is not messenger RNA. There are transfer RNAs, there are ribosomal RNAs, small nuclear RNAs, small nucleolar RNAs, small interfering RNAs, microRNAs, and so on and so forth. In fact, we're learning more and more and more about all the different non-coding RNAs within cells. Non-coding means that they're not messenger RNA. They're RNAs that work and function as an RNA. So the question is, what is a gene? Well, it's a region of DNA whose final product is either a polypeptide or an RNA molecule. And that then now summarizes or completes our look at the mechanics behind how a genotype is translated into a phenotype. Our next subject then is the regulation of this process. What determines whether or not a particular gene is expressed within a particular cell?